Migratory connectivity is a term used to describe the geographic relationship between populations across space and time. For birds, it describes how populations are linked across breeding, migration, and wintering areas. In the prothonotary warbler, a neotropical migratory bird that breeds in bottomland hardwood forests in the southeastern U.S. and winters in coastal mangrove forests in Central and South America, we have a good idea of the extent of the breeding and wintering range, but poor knowledge of how those populations are geographically related. For example, we have been studying prothonotaries in the lower James River watershed since 1987, but have no knowledge of where this species spends the winter. One method of performing a connectivity assessment is to use stable isotope analysis. This technique relies on predictable natural variation across landscapes in ratios of isotopes, which are incorporated into animal tissue through diet. Hydrogen isotopes are commonly used in connectivity assessments because they vary latitudinally. As you can see in this figure, hydrogen values are more positive in the southeastern U.S. and trend more negative as you increase in latitude. Hydrogen isotopes are correlated with precipitation, including water found in lakes and rivers. This analysis is possible because keratinous tissues like feathers are inert and retain the isotopic signature of where they were grown. Most neotropical migratory birds, like prothonotaries, molt their feathers on the breeding grounds and retain them for 1 year. If a feather is sampled on the wintering ground, its isotopic signature can be compared to known values to predict where that bird spent the previous summer. This isoscape is generalized and based on values from several bird species of several guilds. In our study, we seek to build a species-specific hydrogen isoscape to help assign feathers sampled from birds on the wintering ground to breeding origins. A species-specific isoscape is more accurate for assignment because different trophic enhancement factors determine the relationship between hydrogen values from precipitation and actual hydrogen values found in feathers. Our methods will be to collect feathers from across the breeding range from collaborators and long-term monitoring stations and perform stable hydrogen analysis at the Smithsonian Stable Isotope Mass Spectrometry Laboratory to build a breeding ground isoscape. We will then compare isotope ratios from feathers sampled from the wintering grounds using a likelihood-based assignment model incorporating niche modeling as prior information. We will collect feather samples from 10 locations across the core prothonotary wintering range, indicated here in dark green, including samples from collaborators in Costa Rica and Panama, and samples collected on an expedition to Colombia. As I mentioned previously, hydrogen isotopes are correlated with precipitation, including water found in lakes and rivers, which brings us to our second objective. Previous studies have noted that there may be issues with using hydrogen isotopes for species associated with aquatic habitats like wetlands and riparian areas. Processes related to the hydrology of wetlands and the north-south flow of large rivers may result in outlier stable hydrogen values for species growing feathers in these areas. No study has ever been conducted to quantify the potential variation in stable hydrogen for a wetland-associated species, which is why we propose to compare stable hydrogen ratios in the aquatic prothonotary warbler with hydrogen ratios in the prairie warbler, shown here on the right, which is a species that breeds exclusively in upland habitat. We will match locations where prothonotary feathers are sampled with nearby locations where prairie warbler feathers can be sampled at long-term monitoring stations. The mean and standard deviation of hydrogen ratios will be compared for these two species, which will help determine the validity of our connectivity models. Assessing connectivity via stable isotope analysis will pave the way for several exciting analyses in the future. By collecting feathers across the range, we will have access to DNA available at the tip of each feather to study variation in population genetics. Genetic markers tend to vary on a longitudinal axis and can be used to increase assignment accuracy when used in concert with stable isotopes. Our connectivity assessment will also be essential to perform full annual cycle modeling for this species, which will reveal population trends from a holistic approach, taking into account processes happening throughout and across the annual cycle. Additionally, understanding connectivity for this species is important because prothonotaries are considered a species of concern. They have experienced a population decline of more than 40% since the 1960s. Since they are habitat specialists, they're vulnerable to habitat destruction throughout their range. The greatest threat to their persistence is ongoing destruction of tropical mangrove forests. Globally, more than 35% of mangroves have been destroyed since the 1970s, and they face ongoing pressure from human populations. Understanding connectivity sets the framework for hemisphere-wide conservation in this species. Thank you. At this point, I will take any questions.